you very much for coming today. We don't have a large audience, but we have an enthusiastic one, I'm sure. And my script is... My name is Mary Farwell. Some of you know me. I'm the Director of Undergraduate Research in the Division of Research, Economic Development, and Engagement. I'm very happy that you are here and we are here for the first Capture180 Research Challenge competition. Yes, it's very much like the 3MT. You may see lots of parallels. The 3MT is a branded competition that started at the University of Queensland. And our competition is for undergrads, so we've decided to brand it with the help of Carlisle Rogers, who's an indispensable part of our team, who I'll introduce in a few minutes. So we'll get started, but I wanted to do a little bit of thank you. And the first is for Mark Phillips of the North Carolina Biotech Center. We're really pleased that he supported our event, because otherwise we wouldn't be able to have food. So thank you, and take advantage of the wonderful food that we have. Secondly, I want to introduce our judges. I really appreciate their time and efforts. First is Toya Jacobs. She's the Diversity and Inclusion Program Manager in the Office for Equity and Diversity. Second, David Mayo, who's at the Miller School of Entrepreneurship, and Rob Abels in Institutional Technology. No, it's called, is that right? Information Technology and Computing Services, ITCS. So thanks. I also want to acknowledge the visual arts specialist in our division, Ms. Paige Middleton, and Paige designed our programs, which are very, and I think Paige is doing some, she's one of our student workers. Also, one of our student workers is about to come in. She's going to be taking pictures. So we try to have a lot of people helping out. A lot of our colleagues in the division are here. Thank you for coming. The final thanks are the committee. Tom McConnell can't be here, but he runs with Dr. Carlisle Rogers the 3-minute thesis competition. So he and Todd Fraley of the Honors College is here. We basically got together to say we really wanted to do an undergraduate competition. We started with honors students only last year as a pilot. That was successful, so we've decided to brand it and keep it going. Hopefully 8 will be the minimum we'll have, but I'm really pleased that we have 8 wonderful students today. Our last person that helped, Kylie, is back here, and Kylie's going to be taking pictures. And we also have Colby Godwin, who is an undergraduate assistant in our office, and he's ill today, but he did a lot of work behind the scenes. So the program is very simple. Students will stand on the X, and they will present their research in non-specialist terms using one static slide in 3 minutes or less. Students will be presented in the order in the program, and it's a random order. I will introduce each student through the PowerPoint here, and the next slide for each after their names is their static slide that's allowed. Otherwise, they can't have any moving parts. We'll have a timer, that's me, who will warn the students about an hour, about a minute into or 2 minutes into their presentation. So, as you can see on the program, I described the judging. The criteria are comprehension, content, engagement, and communication. So the judges will determine the grand champion winner, and then I will pass out ballots, and we will choose the People's Choice winner based on the balloting. And then finally, so you have some wonderful entertainment and enlightenment, during the judges' deliberation, we have Dr. Tim Christensen, who was the unanimous choice for our speaker today. Thank you, Tim. Thank you, Tim. So without further ado, we're going to bring up our first speaker, and that is Madeline Midyette. And she is speaking in Lead and Bone. Are these on? Yes. Thank you. In Lead and Bone, the Battle of Fort Nyaruka.
Thank you and good morning, everyone. As she said, I am presenting on In Lead and Bone, the Battle of Fort Neoroka. So to give you guys a little bit of background knowledge, the Tuscarora War was a war that was fought here in North Carolina. I am a native of North Carolina, so this is why I chose to pursue this collection. The Tuscarora War was fought between 1711 and 1715, even though we see that the frontal assault of Fort Neoroka was actually fought in 1713. So prior to the onset of the war, we actually had a peaceful cohabitation between the Native Americans and the British, Dutch, and German colonizers. What we start to see is that there were a lot of small tiffs going on between land encroachment and resource encroachment, and the colonizers began to see that the natives were becoming kind of hostile. As anyone would be if your home was being encroached on. So the frontal assault of Fort Neoroka was actually a 3-week siege that led up to a 3-day battle at Fort Neoroka. And what the colonizers did is they got together a number of people. They actually asked for militia from Virginia, and Virginia said, hey, we'll send you down some militiamen if you give us a piece of the Albemarle. So if any of you guys are familiar with that area of North Carolina. We said, no thank you. They went to South Carolina, and the trade-off that they had was actually that if South Carolina sent militia, then we would give them whatever Native Americans they could get their hands on to be sold into slavery. So that was the trade-off between that, which is really interesting to see in the ethnoarchaeology. So the frontal assault of Fort Neoroka, after the 3-week siege that pushed the natives into the fort, was actually ended by burning the fort. So from March 21st to the 23rd, 1713, there were over 300 men, women, and children in the fort, which is what the collection that I'm looking at now currently. So what remains? We have prehistoric ceramics, lithics, bone flint, lead shot, seed and glass, a ton of seed and glass beads from the Native Americans and their cultural remains. So I take a look at that. We also have animal and human remains, which I will be looking at closer in the spring semester. So I will be working with a professor on that. And the research goals, since this is a full academic year presentation, I will be looking at I have been looking at the notes that have been taken by Dr. Phelps, and I'm laying the groundwork for our graduate students who will go on to see the manner in which these men, women, and children died, and the after effects of that, which effectively ended the Neoroka in North Carolina.
Next up, Brinda, Sarathy, Food for Thought, Heart and Soul.
Picture this. You take a bite into a juicy cheeseburger as your taste buds and your stomach are singing the theme song for McDonald's. But have you ever stopped to wonder whether or not your heart was, as the phrase goes, loving it? Here are some facts you probably already know. A high-fat diet is associated with weight gain and fat accumulation in your body, ultimately leading to obesity. Additionally, the consumption of a high-fat diet is associated with something called insulin resistance, where your cells no longer properly respond to the hormone insulin, thus leading to your body not being able to process sugars efficiently and eventually type 2 diabetes. Type 2 diabetes is a serious issue. Over 6% of the population in the US suffers from type 2 diabetes, and over 65% of diabetic patients die from cardiovascular complications. My lab was interested in studying the effects of insulin resistance induced by a high-fat diet in cardiac structure and function, so we studied that in mice this past summer. We had 2 groups of mice, one control group that was fed a standard chow, and one high-fat diet group that was fed a diet comprised of 60% fat. And what we found was astonishing. Simply put, the insulin-resistant mice were having a hard time. They were struggling to have their hearts beat. We found that their hearts were contracting less in diameter with each beat. and they were pumping out less blood with each beat as well. And overall, their hearts were increased in mass. So that really gives meaning to the phrase having a big heart. We then analyzed inflammation and fibrosis, or scar tissue within their hearts, and found that there was a 70% increase in scar tissue, as well as a 40% increase in inflammation, or inflammatory cells known as macrophages. Simply put, the body and their hearts were responding to the diet as though they were being attacked. So, why does this matter? Simply put, diabetic patients and diabetic hearts are more prone to complications such as a heart attack. So, my lab has previously found that the injection of a protein known as Ephrin A1 in the hearts at the time of a myocardial infarction or heart attack has been found to negate those terrible effects of recovery like scar tissue after a heart attack. So we hope to see whether or not we could inject Ephrin A1 prior to a heart attack to find out whether or not that would help diabetic patients in relieving their effects after a heart attack. So I know that cheeseburger sounds amazing today, but I hope that you eat more foods in the future that make your heart say, ba-da-ba-ba-ba, I'm loving it. Thank you.
Next, Miranda Raymond, the phantom protein sequence epidemic. Here you go.
All right, so for those of you who don't know the central dogma of biology, it begins with DNA. DNA is double-stranded, and it encodes all the genetic information. DNA can be then transcribed into RNA, which is single-stranded, and it's different from DNA in that one of its nucleotide bases is uracil rather than thymine. RNA is single-stranded, and it's also found in, or it can be RNA, ribosomal RNA, or messenger RNA. Ribosomal RNA is the structural component of ribosomes, and messenger RNA can be translated into proteins by the ribosome. These proteins are made up of amino acids, and you can see the little circles there. H is for histine, valine is V, and M is methionine. In my project, we used the National Center for Biotechnology's Institute website to use their function called BLAST. BLAST stands for Basic Local Assignment Search Tool, alignment, excuse me. And this can be used to compare nucleotide sequences with their corresponding proteins. The program basically translates an input DNA to a protein for us, so we don't have to let the body do it and confuse us. Basically, it'll do it for us. And so we input the nucleotide sequence, and we searched for proteins that would match this. We came up with all these proteins here, uncharacterized, hypothetical, predicted. These proteins are actually false proteins, and we figured that out by reciprocally blasting these proteins to find their nucleotide sequence that would originally be corresponded with them. But instead of coming up with an mRNA, we came up with an rRNA. rRNAs do not contain the genetic information. They are just structural components of the ribosomes. This is bad because we're actually trying to make results that will lead to future research that we can base our research off of, that can lead to more correct results and further our research goals. But if we have incorrect research and results in our databases, then scientists can predict the wrong results for their future projects. Scientific rigor is very important in the scientific community. We need to have robust and unbiased experimental results. This is clearly not the case with NCBI. It has these problems. These phantom proteins can be found in all areas of life, including animals, fungi, green plants, algae, all areas of life. And that's really bad. We need to fix this problem so that we can have better research in the future.
Maybe that one doesn't work. Our next speaker is Megan Lowry, Can learning keyboarding skills be fun and effective?
In an increasingly technological world, keyboarding skills are very valuable to have. Schools are increasing the percentage of online assignments, homework, and tests, and by the time students are in late middle school, they're expected to be able to type a large amount of text. It's no question that elementary students should be taught the correct keyboarding technique in order to keep up with current standards. The purpose of my research is to test to see if there's any correlation between student satisfaction with a game-based computer keyboarding program and their level of improvement by the end of the year. And we also tested to see if there's any accuracy in their perception of how well they did. The participants in this program were elementary students in a rural southeastern United States city. And there's 2 different schools in this study that we looked at. One was a lower elementary of kindergarten through 2nd, and one was an upper elementary of 3rd through 5th grades. Their abilities were assessed through Typing Test Pro, which basically assessed their words per minute in order to see how well they improved at keyboarding. And at the end of the year, the students were asked a set of 4 questions. There were yes or no questions, and 2 of them we looked at in this study, which the first one is, did you like the keyboarding activities that you did in computer class? And the second one is, do you think you're any better? So the Keyboarding Without Tears program was what we used to teach the students these keyboarding skills, and it facilitated improvement within the students. And this program used pre-keyboarding and keyboarding activities that increased in difficulty throughout the year and depending on the grade level. And so these are 2 graphs that display the results. So the first graph is for the first question on whether or not they liked the activities they did. And it's actually interesting because kindergarten through 2nd actually did better when they said that they didn't like the activities. And it's the opposite for 3rd through 5th grade. They did better when they enjoyed the activities. And the second graph is for the question on whether they think they did better or not. There's not much difference between the younger students, but the older students, they did better. They did better when they thought they did better. So everybody had improvement, but they were more likely to understand that they improved. So the lower elementary school may have had different results because either they didn't understand the questions or they didn't understand their thoughts towards the program at the time they were taking the survey. They got a lot of questions wrong. Like one of the questions was, did you participate in keyboarding games? And a lot of them said no, so they didn't understand the survey. And older students seemed to understand the questions and actually be able to conceptualize whether they did better or not. And they also had more significant improvement than the lower grades, which was expected. And the results give evidence that the older students comprehended their improvement, and this might have influenced their view of the program, or it could be the other way around, that if they liked the program, they did better. More research needs to be done to differentiate these 2 results.
Okay, our next speaker is Amanda Hall. Look who's in charge now.
Imagine this. You're driving down Greenville Boulevard, jamming out to Taylor Swift. As you approach the intersection, the light turns from green to yellow and then to red. Do you continue through the intersection? No, you do not continue through the intersection. Because you perceive the color red and its association to stop, you decided to change your behavior from pressing the gas to stomping on the brake. So what caused this to happen? A visual cue. Visual cues play an important role in behavior and in a social setting. For my research, I'm interested in 2 things. First, how visual cues affect social behavior, and second, the underlying genetics that cause this. To do so, I use zebrafish as a model organism. So up here on the left in the tank, we have a blue striped zebrafish. When you put 2 fish together, they fight. One becomes more aggressive and is the dominant fish. Over time, its blue stripes actually become brighter, while the submissive one, or the less aggressive one, becomes more pale. So in this case, we found that the actual visual cues have a genetic basis for its behavior. So what happens when you take away these visual cues? Up here on the right, we have zebrafish that do not have their blue stripes. Because of this, it causes the two fish to act more aggressive towards each other. Because of this, we want to analyze the underlying genetics in these differences. So this means that you look at the genetics in the fish and you measure it, and they show different quantities depending on if it is a dominant or subordinate fish. So overall, in the end, okay, so next time you approach a red light and you avoid an accident, thank your visual cue of the red light and the stoplight and how it influenced you to stop your behavior.
Next is Sarah Spangler, a clot mess.
Heart attacks, strokes, and embolisms. These are the #1 killers in the U.S. And what do they all have in common? Blood clots, in the wrong place and at the wrong time. When a blood clot forms, it creates this kind of net that stretches to catch and stop the blood flow. These nets are made of a protein called fibrin. Fibrin is thought to have super elastic capabilities. Many studies have studied its structure and the way each unit comes together to form this net. These studies have proven that it has highly elastic properties. It acts like tiny rubber bands being stretched and then springing back into place. We are working to determine which part of the fibrin is actually responsible for this elasticity. Fibrin is made up of 3 smaller units, the alpha, the beta, and the gamma chain. My research focuses on the alpha chain. It's this unstructured region, which is basically a long string of protein without any folds or twists. The alpha chain contains repeating segments, and these repeating segments vary from species to species. Humans have 10, mice have 5, and chicken don't have any. When studies were comparing these species together, it showed that the number of repeats was directly connected to how stretchy the fiber was. The more repeats, the more the stretch. This is the study that we used as a basis for my research. I have been working to mutate fibrin to change its elasticity. By doing this, we can understand what allows fibrin to be so stretchy. This is important to develop treatments for blood clotting gone wrong. We are using a process called protein engineering. We're using this to take the human fibrin and delete sections of the repeated segments. We've made 4 different deletions. We took away sections 1 through 10, 1 through 5, 6 through 10, and cut it all the way before it even started. Once the new fibrin sequence was created, we used bacteria as little DNA replication factories. Then we took that DNA and put it into cells just like ours. The cells acted as protein production factories. We used the cells to see if our new mutated DNA could actually be made into a protein that works. Good news, it can. This is where our research is now. We have our fibrin protein and we're working to purify it. The next step will be testing our protein to see its chemical and physical properties. We'll take each strand and actually stretch it to see how it's changed. Then we can find out how deleting repeats affects the formation of fibers and their elasticity. Who knows, maybe one day our new protein could save your life.
Next, Noah Sonne. And go ahead and pick up. You're going to use over here?
No, I just need this.
Okay, great. Noah, what you didn't know tiny diving boards could do.
All right, thanks for being here. Today I'm going to let you know about a project we did to use tiny diving board structures as a mass detection mechanism. So I want you to imagine a diving board. Now imagine a diving board for an ant. Now imagine a Scrabble tile with about 20 of these diving boards on it. Okay, now take this tile and shrink it down to the point where we can get about 10 of them on the eraser of a pencil. That's the type of structure I'm talking about. So let's come back to a normal scale right now. What happens when I hit something with a hammer? It's going to vibrate. You could think of a xylophone hitting it with a mallet. It vibrates and it makes a really nice sound, right? By attaching these structures to these small diving board structures to another structure, we can actually pull the vibration out of the primary, the bigger structure. All right? So if we look here, this is a measure of the vibration on our primary mass. So this is kind of hard to see, but this is a bigger structure with small diving boards connected to it. Measuring the vibration on the primary mass, we hit it with an impulse, and we can see that the vibration leaves the primary, and it's gone. And then at this synchronization time, the energy is actually returned back to the primary structure. So what happens at this synchronization time? Well, all these little diving boards start to move in the same time. They synchronize, essentially. And they pump the vibration back into your primary mass. Now, if we add mass to every other one of these diving boards, we'll actually see a movement in this synchronization time. So we go from our original synchronization time to half of the original synchronization time by adding a relatively equal amount of mass to every other one of the diving boards. So if we go backwards now and we just measure the primary vibration and we see a change in the coherence time, then we can say, oh, there must be mass added to our system, correct? So by functionalizing each of these opposite cantilever diving board structures to only bind with certain analytes or chemical compounds, we can then say that if we see this shift in the synchronization time, then this compound must be present. And we can then use that as a mass detection or a chemical detection device. And so you could apply this to many aspects. You could think of a breathalyzer that isn't just for alcohol detection, but for other certain microbes or bacteria. And that's what we plan to do. Right now we've shown that it works in the simulations, and our experimental data is starting to agree with the simulations, so that's very cool. I'd like to take questions, but I don't think I have time.
And last but not least, Ankita Mishra, e-cigarettes, yes or no?
In North Carolina, 1 in 5 individuals has used an electronic cigarette. That's 2 million people in our state alone. Since its conception in 2010, the e-cigarette has grown dramatically in its popularity. And research states that most people use them because they think it's a safer alternative than conventional cigarettes. I'm so sorry. Most people use them because they think it's a safer alternative than conventional e-cigarettes. But is this actually the case? In 2014, the CDC found that 1 in every 2 calls to poison control centers was because of e-cigarette misuse. And if this doesn't concern you, the FDA only recently began regulating e-cigarettes in 2016. With the limited research being done on these topics, it's important to understand the effects that they may have on newborns, the most vulnerable population. And that's what my research aimed to do. My research focused on understanding the pregnancy outcomes of patients at the Brody Clinic who may have used e-cigarettes at any point during their pregnancy. The research was divided into 2 components, a retrospective portion and a prospective portion. In the retrospective portion, we looked at electronic health records over the course of a year and tried to identify 200 random samples from that number. From there, we looked at each electronic health record and understood to see if electronic cigarette usage was noted on each record, and also to see multiple health variables that tell us about the health of the newborn. In the prospective portion of the study, we looked at and created a 10-item survey. The survey simply asked patients at the Brody Clinic if they had used electronic cigarettes at any point during their pregnancy, and if they had, why did they use it? Out of 200 surveys that were analyzed in the retrospective portion, 0 surveys indicated that they had used electronic cigarettes. In the prospective portions, 200 surveys were analyzed, and 10 participants said that yes, I did use electronic cigarettes during pregnancy, and I used them because I thought they were a safer alternative. So,
what the results say is that there's a huge discrepancy between what we're seeing in the retrospective portion and the prospective portion of this study. What's being understood is that obstetricians and gynecologists are not asking about e-cigarette use during pregnancy. With the many uncertainties looming around these devices, it's important to be able to educate the patients and make them understand that they are not a safer alternative to conventional cigarettes. There's a lot of controversy behind them. Thank you.
Okay, so this concludes the portion that the students will do, and now our judges will deliberate, and you guys can go out in the hallway, or the lobby, if you will. Would be great. And you need one winner only, please. So, thank you so much. Let's have a big hand for all of our speakers. And now our wonderful speaker will be... We're so happy to have Tim. Tim's, why don't you, Tim's, you're going to tell us a little bit about your background.
Oh, I am? No, my name's Tim. Some of you guys know me. And I've been asked to speak for about 5 minutes. I don't know if I'll do that. I don't have prepared remarks like you guys. But I want to start off by acknowledging a very important person in the room that you guys might not be aware of. Well, actually, you're really aware of this person. This person's name is fear. Right? You guys all feel it. You guys are all here. It's an important motivator in your life. And fear is an indicator of something you should chase, something you should run to. Right? If you're scared of it, it means it's something you have an opportunity to grow with. Running toward fear is increasing your own capacity. I'm not talking about jumping off a bridge with cords, attached to your leg. I couldn't do that. But I'm talking about little things like standing in front of scary people and talking about your research. I run toward fear every day. I am not fearless. I am in fact full of fear. But I try to run toward it every day, and I'm happy to see that you guys are doing the same thing. So you're here giving these 3-minute talks, and you don't know it or not, you don't know it, but you are saving science. Science itself is in crisis in the United States and in the world. It's not that scientists don't know how to be scientists. Scientists are better at being scientists now than they ever have been. They have very specialized language, words, skill sets, all of that stuff. They can do amazing things in lab that people just 10 years ago could not imagine. Scientists have superpowers, but science itself is in decline in the United States. You guys are at the vanguard of solving that problem. And that problem is that scientists have remained in silos so long that no one knows what they're doing, and no one knows that what they're doing is important. We've lost sight as a public that science itself solves problems. Science itself is the most amazing tool that humans have ever invented in the search for objective truth. Now, it's really easy to look at someone who says, you can't argue with my belief, and consider them stupid, because the reason for their belief are facts that are in fact alternative facts. Their beliefs aren't grounded in reality. And we can look at those people and say, oh, they're just stupid. But that doesn't solve the problem. The problem's with us. It's not with them. The problem's with the fact that we, instead of chasing our fears as scientists, content ourselves to talk to other scientists and use our specialized lingo and jargon. We're scared to talk about science with our grandparents that are a little crazy, or our uncle who believes in conspiracy theories. We're scared to talk to those people. But in fact, I encourage you that talking to those people is how we actually solve the problem in science. It's not through science that we solve this problem. It's through being able to communicate our science and being willing, even if we're scared, to communicate our science to a non-traditional audience. Whether it's your grandparents, the crazy person in your poli-sci class that needs some help, or whoever else. You guys are actually building that skill set that will save science. So congratulations. Thank you.
Thanks, Tim. That's wonderful. So, yes, go and he has to go teach. Please help yourself to wonderful refreshments, and our judges will be back in just a few minutes. This is our People's Choice. And this is our grand champion. Okay, so when I call your name, please come up. The first, so exciting. So our grand champion today is Brinda Sarathy. Congratulations. And our People's Choice winner is Sarah Spangler. You're welcome. Thanks so much for coming today, and I hope we keep it going.